Good afternoon, and thank you to all of the organizers of this workshop for allowing me to present on this important topic today. So let's get right to it. Let's quickly go over the learning objectives for this talk. The first thing I'll discuss is what the Office of Study Integrity and Surveillance does in terms of analytical inspections and how we go about identifying potential data integrity issues. Then I'll discuss some examples of data that, while not absolutely indicative of a data integrity issue, are red flags. These are also examples that should allow you, as those who are generating and reviewing data, to know what to be aware of within your facility and operations. Some of the examples we'll discuss include PK and concentration data anomalies in bioequivalence studies, outliers and failures in precision and accuracy assessments, chromatography pitfalls, and software security and audit trails. OSIS is tasked with verifying the reliability and integrity of data generated from the bioanalytical part of a clinical study. In order to do this, we conduct inspections of facilities that perform these bioanalytical studies. And during these inspections, we look at the facility, the staff, previous inspection history. We look at scientific integrity and verified data. We also look very closely at processes and SOPs. We look at data security, and we look closely at the validation of the bioanalytical method. One of the most important pieces of method validation is, does the method demonstrate precision and accuracy? Ultimately, what we have to conclude is, do we have any reason to question the reliability and integrity of the data? So let's get started with an example. Prior to conducting on-site inspections, there's a large amount of data received by the agency that we have access to review, including method validation reports and bioanalytical reports. During this review, if we note potential data anomalies, the goal on-site becomes to investigate these anomalies and, if possible, verify or resolve them. If data anomalies involve PK and/or drug concentration profiles, some of the information we'll ask for is a full PK data analysis of all subjects and the drug concentration profiles for all of the subjects. In this example, I'll show you how PK anomalies and drug concentration profile anomalies can be connected in a bioequivalence study. First, we'll look at a plot showing the distribution of the ratio of the test drug to reference drug for the Cmax across all subjects in a bioequivalence study. And this is an example of a PK anomaly that would cause a red flag. You can see that the TR ratio of Cmax trends above the acceptable bioequivalence range, outlined here in purple, up to and a bit past the midpoint of study subjects. If the study were to stop at this point, bioequivalence would fail. However, then there is a drastic downward trend of the T to R ratio. And by the time we get to the last subject, the overall ratio is 1.03, and the study demonstrates equivalence. Just think about this data while I move to the next few slides. Another red flag is overlapping or nearly overlapping concentration profiles between two or more subjects. This would not be expected in a randomized study population. In the same study referenced on the last slide, you can see from these drug concentration profiles that the data from two different subjects overlaps to an incredibly great extent. Even all the little zigs and zags of the concentration profile are the same between both subjects. And in this situation, you can ask, did this just happen with one subject pair? That could easily be a coincidence. Did it happen with 18 subject pairs? Seems maybe like a bit less of a coincidence. Another question to ask, is there study documentation available to explain the anomalies? Could there have been an accidental switching of tubes during analysis? Conversely, is there documentation present that would indicate the intent to alter results? So are PK and concentration anomalies related? From the graphs below, you can see that the study subjects partition into two distinct populations. Group 1 with a TR ratio of 1.4 and Group 2 with a TR ratio of 0.7, both of which are not bioequivalent. Again, we would not expect a randomized population to partition into distinct populations like this. Moreover, The overlapping subject pairs detected in the previous slide were made up from one subject in group 1 and one subject from group 2. And this could be an indication that the overlapping concentrations are related to the observed PK anomaly. So do these observations mean there is a data integrity issue? The answer is likely yes. First, for the PK results, you would not expect such marked differences between subject groups in a randomized population. And second, the observed overlapping concentrations, again, would not be expected from a random population. This is particularly true if the drug is known to have high intersubject variability. But were the anomalous results done with intention? Not necessarily, and that is a very important thing to remember. A data integrity issue does not mean that it was done with intent. It's critical to investigate and attempt to resolve any data integrity issue. Unintentional errors happen, and if we can confirm an error as the root cause of the data integrity issue, it can be resolved. Conversely, if we verify that there really was an intent to alter data, this could mean the presence of greater issues and call additional data into question. As someone responsible for generating bioanalytical data, what can you do? Look for anomalous trends in the data. Understand what trends would be physiologically improbable. Understand the characteristics of your drug. What would be expected from your study population? For example, a variable versus non-variable drug. And finally, look at the data as an entire entity, if possible, and ask, does it even make sense? So moving on to precision and accuracy. Many times, SOPs allow the exclusion of a specific number of outliers in a precision and accuracy assessment. However, be wary here. If an outlier is not due to a true contemporaneously documented error, for example, double pipetting into a vial, are you actually negating a true inherent variability in the method that could be problematic later? In this example, we're looking at low positive control data, and the value of 135 seen in red was excluded as an outlier. Of note, inclusion of 135 yields an acceptable intra-assay precision of 10.5% for the low positive control. However, inclusion of the value in the percentage inhibition data yields an unacceptable intra-assay precision of 25.5%. And once the value is excluded, precision is acceptable. But food for thought, if the low positive control met the established acceptance criteria for precision with the 135 value included, is it really an outlier? Even more food for thought. The overall inter-assay precision for percentage inhibition of the LPC still failed, even with exclusion of this outlier. So again, this could indicate that the assay truly has higher variability. Another situation we see is where a run fails intra-assay precision for one or more PNA QCs, and then is subsequently excluded from the overall inter-assay precision statistics. In the example shown here, the LQC had an unacceptable %CV or precision value. However, the calibration curve was acceptable, there were no documented errors, and there were no documented equipment malfunctions. However, this run data was excluded from inter-assay PNA statistics. It should not have been, and there was no justifiable reason to exclude this data. Excluding this data skews the true PNA of the assay, and thus the integrity of the PNA data, which is such a critical piece of a method validation. So before excluding precision and accuracy data, ask yourself these questions. Is the data being excluded falsely skewing results or negating true assay variability? And does excluding the data alter the integrity of precision and accuracy results? Moving on to chromatography. Chromatography can be tricky. There are times when a sample should truly be excluded from analysis for poor chromatography, but there are also times when poor chromatography is used as an excuse to exclude or change unwanted data. In the example shown here, and this is a rhetorical question, which of these two samples was labeled as poor chromatography, A, B, or both? The answer, sample B. I had trouble distinguishing why sample B but not sample A was considered poor, so I asked. However, the firm could actually not answer the question of why sample B was poor chromatography and sample A was not, hence a potential data integrity issue. There should be objective criteria in place and used to establish and identify a sample as poor chromatography. A great experiment. Show 5 different analytic chromatograms. How many of the 5 will give you the same answer and the correct answer? Furthermore, we've seen instances where all samples labeled for poor chromatography correspond to a specific sample type, for example, the Cmax. And this is concerning, especially when there are not adequate criteria established for identifying poor chromatography. We get even more concerned when the repeat analysis results look like the graph below. Here, the repeated sample is indicated in blue and is extremely different from the original result and anomalous within the rest of the profile. Multiple samples with this type of change can result in a change of the TR ratios and subsequent change in the overall PK results. Finally, let's talk about audit trails and software security. Software programs used in regulated studies have security measures and audit trails for a reason. And knowing the security features of the software you are using and implementing these features along with the audit trails is critical for maintaining data integrity. Here are a few examples of things to look for. First, is there a record of one analyst with an unusually large number of failed login attempts? You can see in this example there were 7 failed login attempts within 1 hour. The message about a notification being sent, this is actually a default setting within the software, and just because it appears does not mean anyone is actually notified. This feature must be specifically set up to send an email notification to an administrator regarding failed login attempts. But management should be aware of and monitor this type of activity. An occasional failed login attempt due to mistyping a password is obviously expected and happens all the time, but such activity as is pictured here is not. So ask why. Second, in this audit trail, we can see that an unknown sample was used to optimize integration parameters. You can also see that it substantially changed the results. An unknown selectivity sample was used to optimize integration parameters, but there could be potential interfering peaks in an unknown matrix lot, which is exactly what this experiment is attempting to assess. Setting the analyte peak to 0 in an unknown sample can skew results and influence the validation parameters to pass or fail. This is a data integrity issue. And finally, when saving a new results table after reintegration, the file should be saved under a new file name. For example, add a 1 or the letter A after the original name. Overwriting the original integration results does not allow for an assay run to be fully reconstructed. In this example, results were saved 4 times after the initial integration without anyone being able to know what the original 4 results were. Also, note that e-signatures and change reasons were not used or required. These are 2 components of software security that can be implemented to ensure that the changes made were actually allowed. For example, the software can be configured to require a signature for saving a new results table. So now that we've gone through all of these examples, a few knowledge checks. Question 1, which of the following statements is not true? A, in a randomized population, study subjects would be expected to have nearly identical or overlapping profiles. B, values can be excluded from precision and accuracy statistics if there is a contemporaneously documented technical error. C, audit trails are an integral part of ensuring the integrity of bioanalytical data. Or D, clear objective criteria for identifying poor chromatography should be provided in an SOP. The answer here is A. In a randomized population, study subjects would not be expected to have nearly identical or overlapping profiles. In question 2, which of the following data anomalies could indicate a data integrity issue? A, saving a results table multiple times using the same file name. B, inclusion of all data values in precision and accuracy statistics, even when one or two look weird. C, the presence of two distinct PK populations within a random subject population. D, multiple system login failures with no acknowledgement or actions by firm management. And here, there's a bunch of answers, namely A, C, and D. All of these would be red flags and warrant further investigation. So to summarize, the presence of PK anomalies, particularly when they are not reflective of normal physiological responses, may indicate a data integrity issue. Exclusion of data from P&A evaluations should be the exception, not the rule, should be justified with clear objective criteria, and all P&A data should be reported even if it is excluded. Characterization of samples having poor chromatography should be based on clear objective criteria. And finally, understand the software you're using and optimize the security and audit trail functions to ensure data integrity. And again, thank you. Thank you to all of the organizers. I think this is a really great forum, and I appreciate the opportunity to speak.
Thank you for that introduction. I'm pleased to be here today to represent the Division of Pharmacology Toxicology Review within the Office of Generic Drugs to present to you case studies on data integrity issues identified in pharmacology toxicology studies submitted to OGD. Quick disclaimer, this presentation reflects the views of the author and should not be construed to represent FDA's views or policies. Here's a brief outline of what I will cover today. First, I'll provide an introduction to pharmacology toxicology, or as we call ourselves, PharmTox review in the Office of Generic Drugs. Then I will go over observed data integrity issues and the types of studies that are impacted. And then finally, I will go over 3 case studies of data integrity issues. The OGD PharmTox team is consulted by the Office of Pharmaceutical Quality and divisions within OGD when there is a PharmTox safety question for a specific ANDA or drug master file that is referenced by an ANDA. We conduct our reviews within the context of the ANDAs we are consulted on. Therefore, we take into consideration dose, duration of exposure, patient population, and route of administration in our context-specific review. PharmTox review in OGD has similarities to PharmTox review in the Office of New Drugs, or OND. We collaborate frequently on review issues. We apply International Council for Harmonization, or ICH, and FDA guidance. And our goal is to ensure that the generic drug formulation has the same safety profile as its reference listed drug, or RLD. And overall, we operate to fulfill OGD's mission to ensure that high-quality, affordable generic drugs are available to the American public. So what does PharmTox and OGD do? We review the safety of generic formulations where their safety profile may differ from their reference-listed drug. And these differences may be in impurities, excipients, residual solvents, and contaminants from the container closure system. Drug master file or DMF holders and ANDA applicants submit toxicology data to support the specifications of these compounds when there is a question of safety surrounding them. And these submissions generally fall into 3 main categories. We see written justifications, which may include collections of published information or a written expert opinion. Second category, in silico predictions. Commonly, we get QSAR for bacterial mutagenicity, and this is primarily what we accept in the in silico prediction category. And then finally, in vitro and in vivo studies. And these are generally to address genotoxicity or general toxicity through repeated dose toxicity studies. And this is the area where we see data integrity issues. So these in vitro and in vivo, or as we call them, non-clinical studies, are commonly conducted by contract research laboratories on behalf of the sponsor being the DMF holder or ANDA applicant. And as I said before, these are generally to address genotoxicity or general toxicity through repeated dose toxicity studies. And the two most common things we see are bacterial mutagenicity assays, also known as Ames assays, and rodent studies for the general toxicity studies. Now, a unique review challenge to ANDAs is that non-clinical studies are not a standard part of the ANDA submission process as compared to Office of New Drugs, where there is a standard battery of studies that are required to address safety of the drug. We are reviewing case by case these issues and differences of safety profiles, and so commonly we get just a singular non-clinical study as the sole submission to justify safety. Therefore, it's very important that the submitted study is solid and reliable for safety review. One thing to note, non-clinical studies that we see are not always conducted under good laboratory practice or GLP. GLP compliance studies are preferred. We do accept non-GLP studies. However, robust data are necessary. Data integrity issues are not unique to either GLP or non-GLP studies. So what are we looking for? I should say up front that regardless of study type or place of origin, we evaluate each individual study on their own merits. And what we are looking for are robust study data and assay validity. So we evaluate such things as criteria for positive response, use of appropriate controls, GLP compliance, dosing solution analysis, and adherence to standard protocols. We evaluate study design and dose selection. Do the doses tested support the proposed clinical exposure? Are the models used relevant? And we apply ICH and FDA guidances to make recommendations based on safety data. Data integrity is crucial for OGD PharmTox to assess safety. What data integrity issues do we see? We have two main categories. First is suspicious data patterns, such as data repetition, biologically implausible data. We've seen missing information, such as missing data, incomplete methods or results, unsigned study reports, missing pathology reports, claims of GLP compliance, meaning there is a signed GLP compliance statement, but upon review of the study, it was not really GLP compliant. And we've seen unusual data where there's, even between different species and different studies, the same exact data. The second category are false negative results, and these are instances where FDA has data to demonstrate a positive result, but the firm submits a negative result. And these really raise questions about study integrity. So I have these last two bullets here under false negative results, but it really applies to any data integrity issue that we find. These warrant further investigation. It's GLP compliant. We will call for a GLP inspection. And even if it's not GLP compliant, this warrants further investigation by us as well. Okay, time to dig into our data integrity case studies. Case study number 1. This involves a data integrity issue with clinical observations in a rodent study. We received a 90-day oral toxicity study in rats. It was submitted to justify safety of impurities identified in a drug substance, and so this involved a study of rodents that were given the drug substance spiked with impurities. The study had a signed FDA GLP compliance statement. The basic study design involved 5 dosing groups. There was a control group, API group, and then API spiked with impurities at low, mid, and high dose. There was a good number of animals in this 90-day toxicity study, 10 rats per sex per dosing group. In the methods of this study, they stated that they performed clinical observations, and this included 12 parameters, including changes in coat, skin, posture, and excretions. And I mention this because for this particular API, at the dose given to the rats, it is expected that we should see some clinical observations noted, such as diarrhea and hunched posture. But was rather curious that when we got to the individual clinical observations for all 100 rats over the course of 90 days, this is what the data table looked like. All animals were noted as simply normal from day 1 to 90, just like this. This kind of raised a red flag for us with this study. So we looked back at other GLP rodent studies submitted to OGD by the same laboratory, and it revealed the same pattern of observations. All studies submitted contained 100% normal clinical observations for all animals. It's biologically implausible to have zero clinical signs to note across hundreds of animals and across numerous studies. And this really raised some questions for us about the validity of these GLP studies. And so we requested a GLP inspection, which revealed that study personnel at this laboratory were not recording the individual clinical signs that they stated in their protocol. They were basically conducting cage-side interpretation of clinical signs, and this potentially left out data that should have been recorded and placed in the study report. The results of this study, all of these normal clinical signs for all of these animals, really raised questions about study conduct, about potential inadequate drug exposure, and the ability to interpret the data for a conclusion about safety of the impurity was very difficult for us. We ended up not accepting this study. Case study 2, false negative results. As I mentioned earlier, we commonly receive Ames tests, which are in vitro assays that are utilized to assess the mutagenic potential of compounds, and in our case, utilized to assess the mutagenic potential of impurities. A positive Ames test indicates that the impurity is potentially mutagenic and therefore is a potential carcinogen risk because cancer is often linked to mutation. As such, ICH-M7 guidance recommends that potentially genotoxic impurities are controlled to specifically low levels which are linked to risk of cancer. So we received an Ames study to justify controlling an impurity as non-mutagenic in a drug product with a chronic indication. Upon initial review, it was noted that the background plates had low survival and there was a weak positive control response. We were having difficulty interpreting the results of this specific study. Upon further search for information on this impurity, it was revealed that this impurity has actually tested positive in an adequately conducted Ames test. We ended up requesting a GLP inspection of this laboratory that revealed that positive controls were not made fresh, and some of the positive controls used were expired at the time of the study. It was recommended that the study was not reliable for our review. We were not able to accept the study. Case study number 3, data repetition. Here we have another Ames study, and this time this study was submitted to address the potential mutagenicity of an impurity in a drug substance DMF, and this DMF was referenced by 8 different pending and approved ANDAs, meaning that the interpretation of this study could have a very broad impact. Upon review of this study, we noticed an interesting pattern. I have an example here. These 2 tables are 2 experimental repeats of testing mutagenicity of this impurity in strain TA1535. And if you look closely between these 2 tables, I'll just highlight here, there are many repeated individual colony counts. The numbers are very close together, and even the unhighlighted numbers that you see there are actually just switch numbers or even just being off by some numbers. We also noticed that there were all even number colony counts when the SOP stated that they were conducting manual individual colony counts. So we ended up requesting a GLP inspection of this study, which, while we couldn't get an answer for why there was this unusual data repetition and switching of numbers, it was revealed that the employees were not following their SOP. They were not doing individual colony counts. What they did was they counted half the plate and then multiplied by 2 to get the raw data. The study director ultimately failed to assure that all experimental data were accurately recorded and verified. We ended up not accepting the study to address mutagenicity, and this did impact 8 different pending and approved ANDAs. So what is the real issue here? Why is data integrity of non-clinical studies so important? I mentioned earlier that in the ANDA world, a singular non-clinical study is often the sole submission to justify safety of a compound. Here in this example graphic, in these cases where we have a potentially genotoxic impurity, the first line of testing in vitro is the Ames assay. And if a compound is found to be adequately tested in the Ames assay and identified to be negative for mutagenicity, then per ICH guidances, this may allow for potential exposure to higher limits of this impurity, upwards in the multiple milligrams per day realm. However, if this Ames identifies that this impurity is a risk for mutagenicity, then per ICH guidance, this impurity should be controlled tightly down onto the microgram level or even lower. So it's really important that we can rely on these non-clinical studies that are submitted, because the results of these non-clinical studies inform decision on patient exposure to a compound or impurity. So what is the impact of a data integrity issue on these non-clinical studies? If we get a false negative and we don't have our own internal information and we didn't catch that this was not a valid assay or that there was something wrong with this study, this could potentially lead to exposure to unsafe levels of an impurity. So it's really important that we evaluate this study for robustness of data and assay validity to ensure that we are recommending the appropriate exposure level to an impurity in a drug product. So in summary, OGD PharmTox plays a critical role in safety review of generic drugs. Evaluation of data integrity is a crucial aspect of our review. When we identify data integrity issues, we do investigate the scope of them. And ultimately, non-clinical study quality is of utmost importance. Review conclusions of these studies can determine patient exposure to an impurity. Review conclusions can have a broad impact. And when we do identify a data integrity issue and we cannot accept a study, this will result in a major deficiency issued to DMF holders and ANDA applicants. All right, time for our challenge questions. Challenge question number 1. OGD pharmacology toxicology reviews in vitro and in vivo studies to address the safety of which of the following in generic drug formulations? A, impurities, B, excipients, C, residual solvents, D, contaminants from the container closure system, or E, all of the above? The answer is E, all of the above. Therefore, data integrity issues concerning non-clinical studies submitted for a DMF or ANDA can impact numerous aspects of safety evaluation of ANDAs. All right, challenge question number 2. True or false? Non-clinical studies must be conducted under GLP because this is an indicator of reliable data. The answer is false. Many non-clinical studies are submitted with a statement that they are conducted in a GLP environment. However, This does not necessarily mean that the study data are reliable. This concludes my talk today. I want to acknowledge the Division of Pharmacology Toxicology Review Director, Dr. Robert Dorsum, and Deputy Director, Dr. Sruthi King, who helped me put together this talk today. Thank you all for your time and attention. I look forward to answering your questions.
Thank you for your kind introduction. Hello everyone, my name is Byung-taek Oh. I'm very excited to represent the Office of Pharmaceutical Manufacturing Assessment, or PMA, today. From the presentation, we will discuss about role of data integrity in drug applications. Learning objectives. In this presentation, you will learn the impact of data integrity on drug applications, such as why does data integrity matter? What are data integrity indicators by regulatory frameworks? And what will be the cascade impact to bioequivalence study? Then we will talk about data integrity remediation consideration. Finally, we will learn data integrity case study from regulatory submissions. Why does data integrity matter? A quality product of any kind consistently meets the expectations of the user. Drugs are no different. Patients expect safe and effective medicine with every dose they take. Ensuring data reliability is not only GMP requirement, but also key enabler of effective management decision-making. Breach in data integrity, including paper or electronic data, erodes confidence and breaks trust of regulator and public. Failure to uphold data integrity could cast doubt on all information submitted in the application. Data integrity failures could be intentional or unintentional action. However, both are CGMP violation. Data reliability applies across the organization where CGMP activities are being performed. That can be laboratories that develop method or formulation intended to support drug application. Data integrity applies to contract manufacturing organizations for drug substances, final drug products, and even packaging facilities. When systemic CGMP deviations are found by the inspections that implicate the reliability of data generated by the manufacturer, FDA will assess data submitted for drug applications and take appropriate action for those pending approval. Review assessment questions and doubt on all submitted information. Next two slides will introduce potential data integrity issue indicators by regulatory framework. Failure to investigate adequately and document out of specification. FDA prohibits sampling and testing with the goal of achieving a specific results. It means that the firm might perform retesting to achieve passing results without investigation or without scientific justification for the adequate investigation. Several warning letter observations have included observation related to failure to investigate and document out of specification or aberrant results. Testing into compliance. It is not acceptable to use an actual sample in test, prep, or equilibration runs as a means of disguising testing into compliance, such as trial injection or orphan injection. More indicators are described here, such as failure to record activities at the time they were performed. When any data generated by the facility to satisfy a CGMP requirement, all data become a CGMP record. The firm must document or save the data at the time of performance. Another indicator is failure to have controls over electronic data. When login credentials are shared, unique individual cannot be identified. Therefore, the data entry is not attributable. Those are considered data integrity indicators to be in the lookout for during the inspection or while reviewing the establishment inspection report. From previous slides, we learned why data integrity failures are CGMP violations. In addition, we talked about data integrity indicators. Here, I'd like to discuss more about retrospective impact to the drug applications. Previous inspection on Facility A showed good compliance, thus the inspection was classified with the no action indicated or voluntary action indicated. However, recent inspection conducted on the same facility revealed significant CGMP violation due to data integrity failure. Drug application may be impacted by data integrity failure. It would be reasonable to think about what is the extent of impacted data and how systemic are the practices that affect data integrity. Ensuring data integrity is an important component of industry's responsibility to ensure the safety, efficacy, and quality of drugs and of FDA's ability to protect the public health. Industry should assess and identify which applications are impacted by failing data integrity practices. In addition, industry should evaluate reliability of data submitted for review. If the products have been already approved, what is the quality of the drug products distributed to the public? If the regulatory submissions are considered pending applications, is the data submitted in the application reliable? Those are the questions the industry should address when the failing data integrity practices are found. Cascade impact to bioequivalence study. Chemical reverse engineering. This is the method necessary to create a generic version of medication and requires various chemical processes to discover the exact contents of the formula. In addition to being pharmaceutically equivalent, the generic drugs must also be bioequivalent to the brand name drug. That means the generic drug will work in the body in the same way. Data integrity failure may result in the quality of the batches used for bioequivalence studies. Bioequivalence study outcome may therefore be in question if the quality of drug cannot be confirmed. So far, we discussed on retrospective impact on drug applications and cascade impact on bioequivalence study. Here, we'd like to discuss more about the remediation considerations when data integrity failures are identified in the system. When data integrity is identified, FDA requires comprehensive evaluation of data integrity deficiencies. Manufacturers conduct risk assessment of potential effect on drug product quality and application quality and establish corrective action and preventative action. Following slides, we will discuss details of each consideration. Consideration 1. Comprehensive evaluation. Immediate scope should be determined to understand detailed description of strategies and procedures for finding scope of problem. Root cause analysis will perform to explain how and why this breach happened. Then retrospective review may find related products impacted by this breach. List of records, applications, and other documents that have been or will be examined. Therefore, comprehensive evaluation will include determine immediate scope, how and why this breach happened, and retrospective review. And finally, list of previous instances. Consideration 2. Risk assessment. Risk assessment is to understand the impact of data integrity on the drug product quality for distribution, as well as to understand the potential impact on drug product quality and application quality, based on which industry could determine the impact on data submitted in the pending applications. By choosing the proper tool, risk assessment will help mitigate the data integrity failure. It will also help determine the impact on data submitted in the pending applications. Consideration 3. Corrective action and preventative action. Corrective action is to establish action plan based upon root cause analysis to ensure enhanced data monitoring and testing in the future. Industry should take steps to correct unreliable information in the application. Corrective action also includes interim or long-term measures to correct the deficiencies identified during the root cause analysis. Here is an example. 2019 FDA warning letter asked the manufacturer for detailed corrective action plan to ensure the reliability and completeness of all the data. Continue to preventative action. Preventative action is to outline organizational structural roles and responsibilities. The culture determines quality outcomes. The cultural aspects are not overlooked. Habits drive behavior, which in turn drives culture. Breaches in data integrity almost always include the cultural aspect as a contributing root cause. Preventative actions are established to evaluate cultural factors such as management pressure, opportunity, and rationalization. CAPA should be directed to both the facility system and the application data. Solid and appropriate remediation considerations reinstate FDA's confidence in data. Remediation activities include improving the quality management system, culture, and retaining staff. Establishing data reliability is a pivotal for effective decision-making. Remediation implicates both marketed product and those products pending approval. From the previous slides, we discussed about the role of data integrity on drug applications, retrospective analysis, cascade impact to bioequivalence study, and remediation considerations. Here, we'd like to learn data integrity failure, forms remediation consideration, and regulatory decisions from case study. The manufacturer is responsible for drug product manufacturing, and its compliance history showed here. The first inspection was classified with the VAI. Second inspection was classified with the initially OAI, which was downgraded to VAI. And then last inspection was classified with OAI, and warning letter was issued. In the warning letter, significant violations are the site failed to thoroughly review unexplained discrepancies. The site generated false and misleading data. The site conducted preliminary testing prior to testing on the official system. In terms of retrospective analysis, it is good to think about how far back does bad data go. Can you trust the data when the facility initially had OAI, then downgraded to VAI? Or data breach could be found after VAI inspection outcome. Retrospective analysis by FDA identified the earliest finding of unreliable data was identified after first inspection with the VAI classification. Remediation plans. Let's take a look at remediation plans proposed by the manufacturer. The site proposed the following. The manufacturer assessed drug application for data integrity impact. The manufacturer conducted root cause analysis on OSOTs. The manufacturer also submitted their corrective actions as well as internal data integrity audit report for pending applications. OPMA data integrity team has completed the data integrity assessments and determined that the current application data cannot be remediated. The data integrity impact assessment failed to determine immediate scope of data integrity impact and related products, as well as the validity of results. The firm's root cause analysis concluded with no product impact despite invalidation of OSOT, replacement of failing test results without conducting risk assessments, and trial injections. The report has shown lack of justification on why original test results were invalidated and replaced. No detailed investigation reports are available, but only summaries. No evidence that the investigation was thorough and supports the decision to invalidate. Here is a note. Most OSOTs were invalidated due to sample preparation error by technicians or analysts. In addition, trial injections were not addressed by integrity audit. The manufacturer did not assess the presence or impact of trial injections, despite more than 6,000 trial injections were found during the review of laboratory events in the response to the request of additional information. No independent data integrity audit conducted, therefore, the reliability of the report is considered low. Based on the assessment by FDA, regulatory actions on drug applications were taken since the manufacturing facility lacked the necessary controls to assure the data in support of pending applications. The review on the data integrity report has not confirmed the reliability of data generated for applications. We do not think further retrospective analysis will exonerate data given totality of concerns. Submitted data does not reliably depict the quality of the exhibit batches. Without reliable data presented for the review, FDA assessor cannot make a complete assessment. Therefore, pending applications received complete response actions due to the inability to establish the reliability of submitted data, including bioequivalence study. Last, applicants were also asked to use a newly manufactured biobatch to demonstrate bioequivalence to the R&D. Key takeaways. I'd like to reiterate here that ensuring data integrity is an important component of the industry's responsibility to ensure the safety, efficacy, and quality of drugs and of FDA's ability to protect the public health. First, data integrity breaches impact both availability of marketed products as well as the approvability of new products. Second, data integrity breaches results in questioning the quality information for CMC and bioequivalent batches. Third, when data integrity is found, FDA requires a comprehensive evaluation of data integrity deficiencies. Manufacturers conduct risk assessment of potential effect on drug product quality and applications quality. Manufacturer can establish corrective action and preventative action. To make knowledgeable judgment about whether to approve or deny the application, FDA must have confidence that data represented to the application is a true and reliable representation of drug quality. Let's move on to the challenging question. Challenging question number 1. Substantial evidence of efficacy of the drug under review may be questioned when the data to demonstrate the quality of that drug is not reliable. True or false? Answer is true. Data integrity failures may result in the question the quality of batches used for bioequivalence studies. Therefore, bioequivalence study outcome may be in question if the quality of drug cannot be confirmed. Challenging question number 2. Which of the following statements is true? A. Data integrity applies to all sites, including CDMO and CRO. B. The firm may contract with a qualified third-party data integrity consultant to assist in correcting the problems. C. Preventative actions are established to evaluate cultural factors such as management pressure, opportunity, and rationalization. D. Data integrity practices implicate both marketed product and exhibit or biobatches manufactured for applications. E, all of the above? The answer is E, all of the above. Closing thought. Einstein put it powerfully and succinctly. Without changing our patterns of thought, we will not be able to solve the problems we created with our current patterns of thought. Every so often, our ability to see and dissect own problems such as data integrity in the system is very limited by our biases, blind spot, culture, and many other factors. Einstein may highlight critical thinking to solve the problems. Critical thinking is a method of objectively analyzing the data on any problem, subject, or content to form a judgment. By applying critical thinking through objective investigation, root causes can be established, corrective actions taken, and preventative procedures put in place. Let us communicate, collaborate, and work together to deliver a high-quality product that meets the patient's needs. Thank you for your participation today, and I'm now happy to answer your questions. Thank you.
Thank you all for the great presentations. We'll now transition into our final Q&A panel for today. And if you hadn't had a chance to enter your questions in the Q&A chat pod, please do so now. We'll answer as many questions as time allows. To our panel, we'd love to welcome Dr. Partha Roy, who's a recognized senior clinical and regulatory strategist and a proven business leader with 21 years of drug development experience in both US FDA and industry involving new drugs, novel biologics, generics, and biosimilars. Dr. Roy currently leads an FDA CDER office that oversees the thorough assessment of bioequivalence data required to support ANDAs. And we also welcome Dave Coppersmith, who's a senior regulatory counsel in the development of policy, Division of Policy Development, Office of Generic Drugs, in the Office of Drug Policy in CDER. Before joining OGD in May 2019, he was a supervisory regulatory counsel in the Center for Tobacco Products, Office of Compliance and Enforcement. Looks like we have some questions coming in right now, and the first couple of questions is directed to Dr. Nullifer Temple. And here is the first question for Dr. Temple. How often do sponsors disclose fraud or protocol violations for their own studies to the FDA?
Thank you, Ray. So for generic drugs, we rarely see applicants self-reporting falsified data for their studies or reporting protocol violations that jeopardized or could have jeopardized the safety of the study subjects. This type of issues we often see being uncovered either during the inspections or sometimes we learn about them even from the informants, but unfortunately we don't hear much from the sponsors. Having said that, I think more recently we have two examples where it's the applicants who informed us for their own study what appeared to be intentionally compromised data integrity at the clinical site. And I think it's important to talk about them. So let me explain. So in the first case, the applicant reviewed the analytical data prior to the submission. And their review raised a flag for them. So they did their own investigation and provided their evaluation in the submission, pointing out to us why the data from that certain sites were not reliable. And bear in mind, this is prior to either the OSIS inspection finding it or the assessors asking for an explanation during the review of the submission. So it's really encouraging to see the applicants step up and do the right thing. And in the second case, it was after receiving the deficiencies from the Office of Bioequivalence, In their response, the applicants indicated that they investigated the issue and they found integrity of the study conduct at the clinical site had been compromised. And they informed us that the data were not reliable. So we highly encourage the applicants to report the findings of misconduct and falsification that you may discover either during your monitoring or audit or review of the data of your own studies. And that's because individuals and entities that engage in falsification, they tend to repeat this type of a conduct. And we cannot let them continue to jeopardize the welfare of the study subjects or undermine the agency's ability to protect the public health. Thank you.
Thank you for responding to that question. We have another question for Dr. Temple, and here is the question. What is FDA's thinking on using distributed ledger technology to automate data integrity?
Yes, thank you again for that question. I think at this time, we have no experience with this technology, especially for generic drugs. However, I'd say the concept and the application of the DLT are appealing. And with appropriate modification, it can have utility for our work at the FDA. Especially if we think about, say, large clinical trials where the data are synchronized and then there are multiple users across the globe with secured access to a specific network, and they are able to see the records and the data at the same time, including even making any changes to the data or the records. Those can be tracked and viewed at all the nodes within the network. So this technology does seem to have some value. And building such a secure platform by drawing from the concepts of the distributed ledger technology, it could be a futuristic way for sponsors and agencies to perform spot checks or review data in real time and automate the data integrity. So I think this is a very thought-provoking question, and thank you for that. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we received a group of questions for Dr. Shujian Chen. And here is the first question for Dr. Chen. Why should the firm control data integrity on electronic?
Thank you, Ray, and thank you for the great question. As we know from CGMP perspective, electronic data are part of the facility's manufacturing or testing records that are subject to the FDA inspection. Data integrity breach is a violation of CGMP, which often means adulterated drug and can lead to detention under the U.S. law. Therefore, it is the firm's responsibility to implement adequate controls on electronic data to ensure its integrity. On the other hand, from the application point of view, FDA must have confidence that application data, including electronic data, is a true and reliable representation of drug quality. Thus, it will allow the agency to make meaningful decisions on drug approvability. Thank you.
Thank you for responding to that question. We have another question for Dr. Chen, and here is the question. What can firms do to improve their quality culture and ensure data integrity?
Thank you for the question again. We know that firms should implement meaningful and effective strategies to manage data integrity risks. This is stated in the FDA Guidance for Industry on Data Integrity published in 2018. It is the role of management with executive responsibility to create a quality culture that will support data integrity. It is also management's involvement that is essential in preventing and correcting data integrity problems. Without the management support, quality systems can break down and can lead to CGMP non-compliance and violations. where we often observe systematic data integrity issues across multiple sites for the same firm during our inspections. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we welcome Commander Minglei Kui, and here is the first question for Commander Kui. Would you please give some examples of suspicious data or data anomalies which would result in further investigation by the review division, including, but not limited to, for cause inspection?
Thank you, Ray, for that question. The suspicious data are often observed when study failed to meet bioequivalence criteria using the original concentration. And in this case, some decision for reassay for some samples may be made under the code called inconsistent value. And however, this decision for reassay is very biased and very subjective. And often the repeat data or value could change the study outcome. So we consider this type of repeat data as a suspicious data. And for data anomaly, one example would be overlapping PK profiles and unusual PK trend that I showed in my today's presentation. And this type of manipulation can be done by performing an undocumented interim statistical analysis after part of subject PK data become available. During this analysis, for example, if a point of estimate for PK parameters of first half of subjects for the study is particularly high or low, and this suggests that the trial is failing, then this initial analyzed sample can be reanalyzed under new subject identity. And typically with a test or reference treatment switch, this is in order to bring the point estimate close to 1 and make a study pass. So as a result, the point estimate of all these PK parameters for all subjects can be divided into two groups, with the one with the higher value and the other one with the lower value. And the PK profiles, and you often find the PK profiles of this subject in the first group would overlap with some of those subjects in the second group. I hope that's helpful. Thank you.
Thank you for responding to that question. We have another question for Commander Kui, and here is the question. Does NAI or VAI necessarily mean that data is reliable?
Thank you, Ray. That's a very good question. No, the investigator may have data integrity concern, and that need to be further addressed. Even the inspection is not classified as OAI. The OAI non-OAI classification only means that the inspection did not find enough evidence at the study site for this particular inspection to reach OAI level. And however, they could have an inspection summary report. They recommend review division or sponsor take additional steps to assure reliability of data. And those recommendations often lead to further investigation of reviewer division, can have an impact on pending application. And sometimes during the investigation of review division, there could request another for cause inspection. Thank you.
Thank you for responding to those questions. Moving on to our next panelist, we have a few questions for Dr. Cynthia Zhang. And here's the first question for Dr. Zhang. What marketing applications would the BIMO compliance program 7348.003 be applicable to?
Thank you for that question. The BIMO compliance program 7348003 would be applicable for the in vivo bioavailability bioequivalence studies submitted to the agency for the approval of ANDAs, NDAs, BLAs, and biosimilars. Thank you.
Thank you for responding to that question. We have another question for Dr. Zhang, and here is the question. what information during sample collection and processing needs to be properly documented?
Thank you for that question. Yeah, this is a great question. For example, if the samples were collected at protocol-specified time points and within allowable time windows, or if samples were collected outside of the protocol-specified range, deviations would be properly documented and reported in the study reports as protocol deviations. And also, if there's any missing samples, and what was the explanation, and if there's any protocol-specific anticoagulants and or additives were utilized in sample collection and the processing, those information should be documented as well. And if the protocol-specific special handling procedures were in place, then documentation of if those procedures were followed should be also documented as well. Regarding the sample processing, the duration and settings of the sample centrifugation and the time until the freezer storage should be consistent with the specification in the protocol. And again, if there's any special handling procedures, those should be documented. And if there's anything that was not followed, then the protocol deviations should also be documented and the explanations provided. Thank you.
Thank you for responding to that question. Moving on to our next panelist, Dr. Kara Scheibner. Following question came in. Does a data integrity issue always mean that there was intent to manipulate or change?
Thanks. That's a great question. So no, definitely not. There are always unintentional errors that can be made. You can mislabel tubes. There can be an accidental sample switching. There could be cut and paste errors or reporting errors. So things do happen, but just because it's an accidental or an unintentional error doesn't mean that it's okay. It's still a data integrity issue. And the fact that these aren't caught likely means that there's a deficiency somewhere. And so that's why it's so important to recognize and identify potential data integrity issues, to investigate them and try to find a root cause and make the necessary corrections, so it doesn't become something that's a systemic issue and that happens over and over again, even if it isn't intentional.
Thank you for responding to that question. We have another question for Dr. Scheibner, and here's the question. How can data integrity issues be prevented?
So that's another fantastic question, and it's great coming after the one that was just asked. So having very strong, objective processes and SOPs in place that are reviewed often and updated as needed, ensuring that documentation practices are strong, having a strong training program for staff, especially new staff that come in, but also for older staff that might need to have some updates on their training. I mentioned during my talk, know your software and use all the security measures that are available to you. They're there for a reason in these software that are used for regulated purposes. Have detailed investigation procedures, and transparency and trust. Dr. Tompal mentioned during her talk, having involved management. Everybody makes errors, but having an environment where staff aren't afraid to document and report errors is really important for the longevity of any type of data integrity within a firm. Thank you.
Thank you for responding to that question. Moving on to our next panelist, I'd like to welcome Dr. Victoria Keck, And here's the first question for Dr. Keck. What is the benefit of having a GLP compliant study conducted?
Thank you for that question. And compliance is the key word here. For a study to be FDA GLP compliant, that means it follows the good laboratory practices outlined in 21 CFR subchapter A, part 58, also known as good laboratory practice for non-clinical laboratory studies. So what value does that add for a study to be GLP compliant? The very purpose of part 58 outlining non-clinical study GLP practices is to, and I quote the reg here, assure the quality and integrity of the safety data filed. So if a study follows these outlined GLP practices, then we can have a higher level of confidence in the study's quality and integrity. That is why we evaluate for GLP compliance and flag studies that are out of compliance and evaluate the compliance issue. We need to be able to confidently rely on the data submitted. Thank you.
Thank you for responding to that question. We received another audience question for Dr. Keck, and here is the question. Does OGD require submission of SEND data sets for non-clinical tox studies?
And thank you for that question. I noted there are actually a few questions asking about SEND data sets. And at this time on OGD side, there's not a requirement for submission of SEND data sets. However, they may be submitted if they are available. So we will accept them. They're just not a requirement right now. Thank you.
Thank you for responding to those questions. Moving on to our next panelist, we have a few questions we received for Dr. Byung-Taek Oh, and here is the first question for Dr. Oh. Can the sponsor replace drug substance facility where data breaches identified with a new drug substance facility for commercial production?
Thank you for the question. As pointed out during my presentation, it is very important for the industry to evaluate the reliability of data submitted for regulatory review. Submitted data for regulatory review here was generated using impacted drug substances or drug substances manufactured from data integrity failure practices were identified during the inspection. Therefore, it would be reasonable to say that the replacement of drug substance source wouldn't be guaranteeing that original bioequivalence study is considered reliable. Again, bioequivalence study outcome conducted with the original batch or batches may be in question if the quality of the product cannot be confirmed, since the data integrity failures may cause a question the quality of batches used for bioequivalence studies. The applicants might be asked to use a newly manufactured biobatch to demonstrate bioequivalence to RLD. Thank you.
Thank you for responding to that question. We received another question for Dr. Oh, and here is the question. Will API manufacturers be audited if their DMF is referenced in any ANDAs, BLAs, and NDAs?
Thank you for the question again. Once again, I'd like to highlight that the data integrity applies to all manufacturing or testing activities. It is for sure including API manufacturer here. If the drug master file is referenced to support regulatory submissions such as ANDAs, NDAs, and BLAs, it is obviously subject to CGMP regulation. In other words, the manufacturer in DMF such as API manufacturing facility and testing facility will be considered under CGMP regulation. Therefore, the facilities will be evaluated and inspected by the agency. Thank you.
Thank you for responding to that question. We have a few questions. Moving on to a new panelist for Dr. Partha Roy. And here's the first question. Please explain why data integrity is a big concern. We only hear just of 1 or 2 CROs committing fraud.
Thank you for that question. As all my colleagues here have already stated several times in their presentation, that data integrity is at the heart of the accuracy and the reliability of the evidence that is needed for our regulatory decision making. FDA expects all data, I just repeat here, all data collected and derived to be reliable and accurate. Ultimately, data inaccuracy and discrepancies prevent us from ensuring the safety, efficacy, and quality of drugs and FDA's ability to protect patients and public health.
Thank you for responding to that question. We do have another question for Dr. Roy, and here is the question. How can we inform the FDA of fraud by CROs during the study?
Yeah, that's a great question. Obviously, you can write to the FDA, email, phone calls, but I think the most convenient way, an easy way to report data integrity issues to FDA would be to use the following emails: druginfo@fda.hhs.gov and genericdrugs@fda.hhs.gov. I think all those two emails are in our website, so you can look it up there. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we have a few questions for Dave Coppersmith, and here is the first question. Does FDA plan to issue guidance with recommendations on data?
Thank you for the question. And so the answer is yes. As noted by several of my colleagues during their presentations and also by Dr. Chen earlier in this panel discussion, back in 2018, FDA issued a guidance for industry titled Data Integrity and Compliance with Drug CGMPs, Current Good Manufacturing Practice, which clarifies the role of data integrity in CGMP for drugs and also includes general recommendations regarding data integrity. In addition, FDA is also planning to issue a guidance for industry with recommendations on data integrity for bioavailability and bioequivalent studies at testing sites. The availability of that guidance will be announced with a notice in the Federal Register once the guidance is published, and that notice will also identify a comment period during which interested parties can submit comments regarding that guidance. Thank you.
Thank you for responding to that question. We have another question for Dave Coppersmith, and here is the question. Does FDA share or obtain information from other regulatory?
Yes, thank you for that question as well. So FDA does have a mechanism for sharing non-public information with foreign regulatory agencies. Specifically, that mechanism is a confidentiality commitment, which is a document that establishes a legal framework that allows, but does not require, FDA to share certain kinds of non-public information with FDA counterparts in foreign countries and international organizations as part of regulatory activities. Thank you.
Thank you for responding to that question. Moving back to the beginning of our panel, we received some questions for Dr. Nullifer Tample, and here's the question. Thank you for clarifying the expectation on the role of the sponsor in ensuring data integrity. When a CRO is identified to be involved in data integrity issues, for example, for a clinical study, excluding bioanalytical, conducted at that site, what is expected from FDA to honor sponsors' due diligence study monitoring report as justification to demonstrate no observed integrity issues for the clinical study in question?
Hi, Ray. Thank you for the question. So if I'm understanding this question correctly, I assume that the data integrity concern is coming from the agency, means it's been raised by the agency, which then would mean it's either from an inspection or through a deficiency letter from the Office of Bioequivalence asking to explain or address the data integrity concern. So if it's during the inspection, our investigators, they provide sufficient opportunities both during and after the inspections to kind of respond to the concerns that have been observed and raised during the inspection. And whatever the response that is received, it is assessed not only by OSIS, but also by the Office of Bioequivalence. So there will be an assessment done. And similarly, if the Office of Bioequivalence communication, if it's coming from there, we would request for a response with supporting data and information. Generally, that type of a language is used as a standard. So if it's in your judgment that the monitoring report could serve as your supporting document, you should submit it and it will be reviewed. Now, whether that will be found adequate will depend on the thoroughness of the monitoring and the investigation performed, and also the relevance to the issue and the discussion. Because ultimately, we need to have confidence for accepting the sponsor's conclusion of no data integrity issue after reviewing all the information that is provided to us. And I'll give you an example again. We have seen reports telling us that the sponsor or the third party did the investigation for a subset of samples and found no issues at that site. However, that subset did not include any of the very samples for which the agency had determined that the data were not reliable. So the relevance and the scope of the investigation and your monitoring is important. How encompassing it is plays a role. Thank you.
Thank you for responding to that question. Moving on to the next panelist, we received a question for Dr. Shujun Chen. And here's the question. Please discuss DI issues identified in the manufacturing operation, excluding QC lab.
Thank you, Ray, and that's a great question. As discussed during my presentation, during both CGMP inspections and during application review, DI issues can be identified for the manufacturing operation, also in the submitted batch records, in addition to the often observed testing DI issues. In terms of the manufacturing DI issues, those can include access control issues, such as granting the operators administrative privileges and allowing them to modify the electronic batch records without QA approval. Sometimes we can see that there are data recording issues, such as not recording activities contemporaneously and backdating. We also observe data falsification issues. Some examples include entering incorrect sample weights, or sometimes incorrect process parameters, as well as investigation-related DI issues. Examples include not recording or adequately investigating a deviation or OOS in-process test results. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we received another question from Commander Minglei Kui. And here is the question. Is clinical monitoring a regulatory obligation for BE studies, and can we adopt risk-based monitoring and only monitor critical data?
Thank you. That's a good question. Yes, clinical monitoring should be applicant and sponsor's responsibility. Applicant or sponsor, they needed to ensure that study is conducted and the data is generated, collected, recorded, and reported in compliance with the regulation, and to ensure the reliability of the data. So risk-based approach can help guide appropriate allocation of resources to important process and procedures. And so applicants, sponsor are expected to develop a quality management system using risk-based approach to manage quality throughout all stages of clinical and analytical processes and data lifecycle. Thank you.
Thank you for responding to that question. We just received another question for Dr. Victoria Keck, and here is the question. You mentioned that in silico prediction for bacterial mutagenicity is an accepted safety justification. Would OGD accept in silico prediction for general toxicity?
Thank you for that question. The answer is no. The only safety endpoints that we recognize as having been adequately validated in silico prediction models is for bacterial mutagenicity. To that point, even ICH recognizes this. In silico prediction of bacterial mutagenicity is discussed as an option for assessing genotoxicity of impurities in the ICH-M7 guidance for mutagenic impurities. And it is the only in silico method mentioned in the impurity guidances from ICH. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we did receive a question for Dr. Partha Roy, and here's the question. From working in industry, I'm familiar with data integrity. However, I rarely hear about recalls stemming from data integrity violations. However, I do hear about adulterated drugs. Are we going to start seeing recalls where data integrity is clearly identified to the public?
That's a great question. Thank you. So the real answer is really depends. Recall decisions are taken very, very seriously within the agency and really not taken in a vacuum. That decision is not based on just one source of information. It is often a multifactorial decision making. Hence, quality culture, as stated by my colleagues, is an important parameter to establish. Ultimately, if data integrity issue rises to the level of reasonable and significant evidence of loss of efficacy and or significant impact on safety of drug products, such recall is certainly a possibility. Thank you.
Thank you for answering that question, Dr. Partha Roy. It looks like at this moment we have gone through all of the submitted questions relevant to the presentations at this time. We'll give the audience just a moment to see if any more questions come in that we can quickly review. And we'll just take a momentary break to do that. We'll be right back. All right, we wanted to give everyone another chance. That seems like that's all the questions that have come in at this time that we're able to answer based on the presentations. I may see something coming in just a moment. I think that's about it. Well, we want to congratulate and thank everyone who's presented today and sat on our panels for great presentations and answering numerous questions that came in. We'll start day 2 of the Generic Drug Forum tomorrow at